EVA is the European Association for Vision and Eye Research. It's a European organisation which aims to foster research in vision and ophthalmology, encouraging both young and established researchers and encouraging a dialogue and an exchange of information. It was established as an entity in 1997, but preceding that there was an umbrella organisation of European vision research associations loosely joined together, which held joint meetings for a number of years, and it arose out of that. Its first meeting was in 1998. We hold our meetings once a year, and we encourage researchers to come to present their work at special interest symposia and free paper and poster sessions. We're particularly interested in bringing basic scientists and clinicians together with an increasing emphasis on translational research. The aims are to foster research, and for that we also have an EVA Foundation, which is important. This has been in existence now for 3 years or so and established in order to promote research and to promote the funding of fellowships for young researchers, as well as to promote new initiatives in research that will be useful in the next coming years. It is a challenge. In Europe there are many ophthalmologists. Different countries will organise their systems quite differently. In some countries, private medicine means that many ophthalmologists do not have associations with a university department, but in some countries the system is organised much more centrally. For example, in the UK, the National Health Service is able to give us quite a lot of input into academic ophthalmology, so that most researchers will have an academic appointment at the university as well as in a hospital. When it comes to the vision scientists, who we're really keen to work with, there is clearly an issue about funding vision research, which is not at the forefront perhaps of European funding at the moment, and we'd like to encourage it to increase its impact. And so we are very keen to encourage young researchers to stay in vision research and to enter the field. The European situation is that there has been extremely good vision research done with some extraordinary discoveries in the last 20-30 years. And the highlights would include perhaps the seminal work on the contact lens, the discovery of, and so forth of the contact lens materials and development of contact lenses, cataract surgery techniques, corneal surgery, techniques involved in treatment of macular degeneration, across the board. But essentially we're aware that EVA needs to be a forum for the people to come and present their research. We'd like to encourage them to think of it as the first port of call for their European research.
Efficiently funded.
So the first thing to say is that, of course, the EU isn't the same as the United States. So that actually the EU is an umbrella, but we have funding at a national level in all the nation states. So at a national level, of course, it varies tremendously from country to country. Most countries have some form of National Medical Research Council, which on a national level takes the sort of position that you would ascribe to the National Eye Institute for the funding. On an absolute European level, of course, there is EU funding, with calls going out to establish networks and partnerships and EU-funded projects which bring people together. There has been increased funding over the years, but we feel that the funding for vision is still something that needs more lobbying in the context of improving our profile with respect to neuroscience, for example, and other areas. So I work on mitochondrial optic neuropathies. These are conditions in which there are genetic abnormalities affecting the optic nerve, and patients lose vision in a number of ways, but ultimately it results in the loss of the nerve cells of the optic nerve, called the retinal ganglion cells. I've been working on this for a number of years, initially working with families to identify the gene, and identifying the gene called OPA1 a number of years ago. And our group was the first to publish this alongside a French group in 2001. This work then has led to the work of trying to establish what the mechanisms of the disease are, the so-called pathophysiology, and how the mutation in the gene can result in a dysfunctional protein and what that then does to the retinal ganglion cells. This has involved work in the laboratory, in cell culture, and with model systems, as well as with patients, trying to understand the natural history of the disease and the so-called phenotype, which is the appearance of the patients, and genotype, which is the genetic defect, and how they could be correlated. And the ultimate aim of this work now is beginning to slowly come to therapeutic intervention. This is now at an early stage. Still, there are no effective treatments for mitochondrial optic neuropathies, but there has been a recent increase in interest in novel pharmacological therapies, and this has led to some clinical trials. One stands out particularly, which is the clinical trial of idebenone, which is an agent that has potential to improve mitochondrial function and may have some place in patients with mitochondrial optic neuropathies. Well, when we study the mechanism of ganglion cell loss in the mitochondrial optic neuropathies, and the particular one that I work on is dominant optic atrophy due to mutations in the OPA1 gene, we have to be aware of reactive oxygen species, or ROS, because they are a product of mitochondrial function. They have signaling mechanisms and roles in the cell which are quite important and beneficial, but they can, in excess, of course, be damaging. So some of the therapies that are being proposed are broadly antioxidant in their description, although that may not be the only way in which they can act. So a combination of neuroprotective and antioxidant pharmacological therapies may well prove to be the way that we will be able to treat these in the future, although this is clearly at a very early stage. The answer to that is much more complex, but it's clearly a balance, and there's in different tissues potentially different capacities to both generate and mop up reactive oxygen species that will be quite important in the way that the disease evolves, and specifically in a disease where ROS is thought to be of importance. I think for the first time we're starting to see globally an interest in rare diseases, and this is of great importance because rare or orphan diseases have been neglected by funding bodies for many years. So we're seeing a sea change in the way that that's perceived both at the EU and perhaps worldwide. Patient associations are becoming increasingly aware that rare diseases could be treatable, and they've always been a group of diseases which potentially have no therapies because they're genetic or very complex. This is now leading to the fact that there are people trying to investigate pharmacological interventions as well as new therapies, which might include stem cell therapy or gene therapies for both retinal diseases, which could be genetic, or optic nerve diseases. So I think we are seeing a new era perhaps of translational research now, potentially more focusing on pharmacological interventions in the mitochondrial optic neuropathies. But that is not to say that there wouldn't be some role for gene therapy, for example. Well, clearly in the retinal degenerations and retinal dystrophies that are genetic, such as retinitis pigmentosa and the macular dystrophies, there's been very exciting work done recently, both on gene therapy, for example, and stem cell therapies, which are in their early stage but clearly have some promise. The techniques that are being developed may be of more applicability to other parts of the eye and to other diseases of the eye. So there's cross-fertilization there. There's also cross-fertilization between the clinicians who are working with patient groups now and can categorize them genetically very accurately, and the basic scientists who can then focus therapies on specific cellular pathways. And so I think that one can't exist without the other. The basic scientists, the vision scientists, and the clinical scientists really do need to talk to each other. So genetic testing falls into several areas, and some of which are not really controversial at all, and some of which potentially are controversial. So genetic testing in the diagnosis and management of a patient who has lost vision due to a genetic disease is probably not particularly controversial. The patient is carefully counseled and desires to know what the genetic defect might be, and if we know what the genes are that could be involved, we can test that patient's DNA from the blood and come up with a genetic molecular diagnosis. This can be very helpful for the family in both deciding whether the disease is severe enough for them to contemplate not having children, or to contemplate early school interventions if their child is affected, and all sorts of decisions that families need to make. On the other hand, rarely do we see in eyes diseases that are so severe that people choose not to have children, and so that we don't face that decision-making process very often. But we work very closely with our clinical genetics colleagues, because where we are dealing with families with children born with very severe handicaps due to multiple systemic disease, we need a lot more input from the clinical genetics for counselling. We don't, in the ophthalmology sector, really tend to have diseases that lead to a lot of requests, for example, for antenatal testing, although that is certainly possible in some diseases, such as the anterior segment dysgenesis, aniridia, and so on. And this is certainly done, but it's a small part of our work, really, in genetic eye disease, a very small part. So this is a paradox, because I feel that my whole reason for being interested in genetic eye disease is to be able to genetically test patients for specific problems, to help them make decisions, advance diagnosis, manage their condition, and then potentially even help with therapeutic intervention. Having said that, the genetic testing of people by the internet, essentially through companies that market these tests, presents a lot of problems. And if it's done in the context of individuals who have no genetic knowledge, it's potentially going to lead to huge and quite fundamental problems. If the testing is normal, clearly there isn't a problem. If the testing is abnormal, those patients will clearly not understand why it is or what the implications are. That is not to say that they don't have the capacity to go away and read, but most patients don't have the time and the training to read the relevant literature. They will need to be seen by a professional. They need to be entered into the system for counselling. And actually, when it comes to the genetic testing for a lot of predisposing risk factor genes, we don't actually have the knowledge to tell them what that will mean for them individually. And so it's opening a Pandora's box, which isn't particularly helpful at this stage. For example, there are some genes that you could predict an individual's increased likelihood of having a heart attack. Hypercholesterolemia is a good example. And in that, we do the testing in our healthcare systems because we feel it's helpful to give them this advice, and there are some concrete ways you can measure the endpoints. But in others, there will be very, very difficult data or very little data to suggest what the risk factors will actually mean to the individual. They're not then able to take any action on the basis of this information. They can't really modify their lifestyles, they can't change their basic risks, and therefore it's not in any respect a helpful test. And not only that, but even with genes that cause disease in some individuals when they are mutated, we know that the severity of the disease can be extraordinarily diverse, from virtually unaffected and mild through to very severe. So having a genetic test that does not predict whether you will be severely affected or mildly affected, and therefore, again, the issues about counselling and advice and what the implications are enormous. So at this stage, it's something that the internet has essentially foisted upon us, and it's an incredibly exciting and interesting time, but as far as patients and the management of patients is concerned, it doesn't necessarily improve healthcare. EVER is a society that's small and young, and we have 1,000 delegates this year. The membership is composed of both clinicians and scientists, and we're particularly passionate about the fact that at the meeting we encourage people to socialise, so much so that we will invite them to a welcome reception and a farewell cocktails, and we build into our agenda dinners and coffees and social occasions. And we feel that that's a particularly important European contribution to this type of meeting, because people have commented many times that this is a really good forum for the exchange of ideas, to meet new people, to be introduced to the big people in the field in a way that you may not have that opportunity if you were attending a meeting where there were no social interactions.